The overall goal of this procedure is to minimize degradation of biological images caused by the air spaces in plant leaves to acquire clearer images of living cells within the leaf. If the leaf is mounted in an aqueous mounting medium and a fluorophore is excited by a laser, many unfilled air spaces and changes in refractive index within the tissue, shown in orange, limit the strength of the signal deep from within the tissue in the resultant image. If the leaf is instead mounted in perfluorodecalin, or PFD, the resulting laser excitation of the fluorophore is less attenuated and allows better transmission of emitted signals. Ultimately, use of scanning laser confocal microscopy with the PFD mounting medium gives clear images of living cells within the plant leaf at a depth that is more than double that possible when an aqueous mounting medium is used.
The main advantage of this technique over existing methods like fixation or mounting leaves in aqueous media is that it reduces refraction within the mesophyll and enables more accurate imaging of living cells deeper within the leaf, which has been hitherto problematic.
This method can help answer key questions in plant science, such as addressing mesophyll cell biology and signaling in vivo, the colonization of leaves by pathogens, and pathogen-mesophyll interactions.
Though this method can provide insight into fundamental plant biology, it can also be applied to other highly refractive tissues that are rich in air spaces, such as insect spiracles or the vertebrate lung.
Demonstrating this procedure will be Dr. George Littlejohn from my laboratory.
To mount leaf samples in PFD,